Hello, and this is 8B. This is going to be talking about Mendel and genetics and with using Punnett squares. Mendel's an Austrian monk from way back when that actually looks around. He's a farmer. Remember, monks have lots of free time. He's a farmer. He's actually in charge of helping grow food for the monastery and probably the villages that are around the monastery. But anyway, he starts looking at pea plants and he starts noticing that they're can be rather strange. So he comes up with the idea of heredity, where traits are actually passed from one generation to the next. He actually starts looking at these pea pods and he sees there are 7 different characteristics that he can see that are different. He sees the seed, the form, whether it be round or wrinkled. He sees cotyledons, whether these are the two leaves that pop out of the seed. Actually showing you the dicotyledon or the monocotyledon, but this is the dicotyledon. And they actually come out as yellow or green. The flower can be white or violet. The pea pod can be smooth in terms of form, can be smooth or constricted, where you can actually see the individual peas. Color can be yellow or green. And then the stem, we can actually have the flowers at the axil, which is sort of like at the crux of the branches, or we can have it actually at the terminal at the end of the branches. And the size, we can have a plant that's called tall, anywhere between 6 and 7 feet tall, and we can have short plants that are less than 1. And he started seeing these things pop up in pea fields. So he started wondering, are these things actually genes that travel from one generation to another, that it can actually affect the offspring and can be predicted? And so he actually sets up some things, and the scientific community took about 30 years to catch up to Mendel's work. So he's using terminology that we still use today. We do call this the parent generation. We call these the filials, filial 1 and 2. We have dominant and recessive. We have his 3 laws, which we'll talk about. We still use Punnett squares. I'll explain what a Punnett square is. But the parent generation, if we look at these 2 adults here, we have 2 homozygous. I'm not sure who the mom and the dad is, and what he would do is he would actually cut off the male parts of the flower of the one he wanted to be female, and he'd cut off the female parts of the flower he wanted to be male, and he would cross pollinate these plants. So he had a mom and a dad, and I don't know whether the mom, um, I don't know whether which one of these was the mom, but one of them he actually used to get the pollen, and the other one he actually used to have the seed. Okay, so he took those and he crossed those homozygous tall and homozygous short, 6 to 7 foot or 1 foot. Looks like everything else on there is probably the same. He got down to the first filial generation. He calls this the F1 generation. The offspring are all going to be heterozygous. They're all going to get a tall one from one of the parent, and they're going to get a short one from the other parent. And then he crosses those, and he actually finds out that he's got 3 talls and a short. But he actually figures out when he does this is he's got 1 homozygous tall, 2 heterozygous tall and a short, short being masked over, and then he's got a short one, which is homozygous short. So he's actually coming up with this terminology, and he finds out that he can actually predict how plants are going to go, and he does that with all 7 characteristics. So when we actually start talking about this, we have to start talking about a little bit more terminology. Gene is the section on the homozygous, or the homologous, basically they're the section on these sister chromatids that actually code for the same thing. The allele is actually the 2 different types. So we actually have purple and we have white. I was going to say pink. I wish they would have put white. And if we actually give these symbols to it, we'll actually say that this one is the dominant and this is the recessive. This is the one that shows up most often, most common. This is the one that shows up every once in a while. And we have different examples. We can have plant examples and we can have animal examples, or in this case, human examples. Plant examples like what Mendel was saying, where color of seeds, shape of seeds, form of the pea pods, color of flowers. We can't really see how tall. We can't see where the flowers are placed. We can see that the seeds at least look like they're all round. We can see that they're all green. Cotyledons, we can't tell. And then animal examples, or in this case human examples, where we can talk about eye color, skin color, hair color, freckles, lack of or presence of widow's peak. We can talk about all kinds of things. Okay, Mendel comes up with 3 laws. First one's called the law of segregation. And basically what you do is you have these sister chromatids, homologous chromosomes, and we actually have these sister chromatids with 2 different alleles, 2 different genes that code for a different thing. They're called alleles. We have a dominant one, we have a recessive one, and that they separate through meiosis. So if this was a sperm and this was an egg, we'd end up with the same type of alleles. And if we actually had this one as a sperm and this one as an egg, or this one as an egg and that one as a sperm, we'd end up with different alleles. Law of independent assortment. Traits are inherited independently. You can really have, let's say a plant has 4,000 kids, baby plants, and 3,000 may be this one, and then 500 may be this one, and then 1,500 may be this one. We actually, you can't tell how it's going to work out. It's all independent. It's like flipping a coin. So you can get this one, and then you can get this one, and then you can get this one, and 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 this one. It's independent. And then the rule of dominance, we have the dominant alleles which mask over the recessive ones. And that's what we actually show with uppercase being dominant and lowercase being recessive. So let's talk about dominant and recessive. These traits actually mask over the recessive. So when we actually have the big T and the little t is the one that shows up. So they're all going to be tall. Even though these are heterozygous, they actually do have a large dominant percentage, so where they all actually end up being tall. And the recessive trait, this is a trait that's covered over by the dominant, and it's only visible unless it comes in with both alleles, in case of like this TT over here, where we get a short plant. So dominant always masks over recessive. If they're both dominant, they're purebreds, they're homozygous. If they're both recessive, they're purebreds, they're heterozygous. If they have one of each, the dominant one is going to show up. Those are heterozygous. Here's examples of some traits that people can do. Rolling tongues, which is dominant, most people can do it. Tongue folding, this is inability dominant. I'm not exactly sure what that means, but that means you can take your tongue and you can bend it without using your teeth at the top of your mouth, like that. Left over right thumb when grasping, left, that's dominant. PTC, this is a chemical that you can put on paper and put it on your tongue. Most people can test it. Most people have oval faces versus square, and they're not really square. Cleft chin, no cleft chin is dominant. Middle digit hair, this is actually on your fingers. That is, and I think that's dominant. RH factor is dominant. That's like saying A positive, A negative, rather than just saying like O. Eyebrow shape, separated is dominant to joined. Very few people have single eyebrows going across their face. Eyelash length, long is dominant. Hitchhiker's thumb, that one is dominant. I'm sorry, that's recessive. Most people are this way, and I can't imagine that because every time I look at my thumb, it's the hitchhikers. Dimples are dominant, almond eyes are dominant, recessive, autosomal dwarfism and albinism, and then here are dominant traits, and you can see there are a whole bunch of them, and you can pause the video and take a look at it. And here are some recessive traits, and I'm not going to go over all those because I don't want this anything longer than it is. So homozygous for both alleles are the same information. They can be either dominant or recessive. And they're also called purebreds. And heterozygous is one allele, one kind, one capital, one lowercase. Like this one, dominant's always written first. Those are called hybrids. This would be a dominant set of alleles on 2 sister chromatids, and this would be a non-homozygous pair. This would be a heterozygous because we have one of each. And then we can work with genetics, and we actually say that this woman is a carrier because this is a bad allele. And luckily the good one is dominant, and this is also a faulty carrier for the father because he's got a bad allele. And we have eggs and sperm, and we have independent assortment. So this normal egg could actually go for all of her kids, and this abnormal sperm could go to all the kids, and they'll all appear as carriers normal. Or we could actually get one of these. I hate that when that happens. We can get one of these and one of those, like in this case. So out of this assortment, if we just did a Punnett square, we would say there's a 25% chance of having a non-carrier baby. There's a 50% chance of actually having carrier baby, and then there is a 25% chance of actually having an affected baby. But this doesn't mean you're going to have 4 kids and you're going to get this. You have a 1 in 4, a 2 in 4, a 1 in 4 every time you have a kid. Phenotype and genotype. Phenotype is actually how the trait appears in the individual. It's the blue eyes, it's the brown hair, it's the tall, it's the pink flowers, it's whatever you can see with your eyes. It's how it's appeared. The genotype is the letter combination, and this describes the phenotype. So this is writing the A's and the C's and the T's and the G's. Why did I just pick those letters? It's like the P's and the Q's and the R's and the Y's that we actually use to show what is going to describe the phenotype. So freckles, this young boy actually has quite a few freckles, and that'll show up as either being one from each of the parents or one dominant, one recessive. Which of these is the pheno, which is the genotype? Phenotype and genotype. How about for blue eyes? Phenotype is blue and genotype is the BB. And then what about for widow's peak? And this is Eddie Munster. If you ever watched the Munsters from old time TV, it's when I was a kid. He had just the epitome of a great widow's peak. It's not normally this size, but the widow's peak is the phenotype, and the genotype is either the W, homozygous, or heterozygous. And you notice this one actually is a homozygous. The only time you get blue eyes is if you get both recessive traits. So which are purebreds? Purebred, hybrid, purebred, purebred, hybrid. Punnett squares. Punnett squares are actually named after those small little baskets for strawberries and raspberries and blueberries and whatever kind of berries you want to put in there, but that's what they're named. And you can imagine 4 of them, so they were called Punnett squares. This is a diagram used to predict the outcome of a particular cross or breeding experiments. You can do this if you're actually raising dogs. You can do it if you're raising peas. You can do it if you're raising kids. So mom we usually put on this side. Dad we usually put on top, and then on the outside you actually represent the parents' alleles, and BB, that's heterozygous brown eyes. Brown is the dominant one, and mom is blue eyes, so she's got to be homozygous for blue. Purebred, hybrid. And then what you do is you put the different alleles inside the box, or inside the circles, or right by the box. And now we just say, in this box, we're going to get this allele from mom and this allele from dad. And then that's going to be brown eyes. I don't know which one we go to next. I'll let the first one pop up. Down here we get this one from dad, this one from mom. Another set of brown eyes. This one from dad, this one from mom. Blue eyes, and this one from mom and this one from dad. And it doesn't matter if they are the same, like these homozygous recessive. It doesn't matter how you write them. You can't tell which one of those is mom and dad. This one you can tell, and you always put dominant first. So this will be BB, and that'll be another set of blue eyes. So you have a 50% chance of getting brown, 50% of blue, but you may have 8 kids and they're all blue-eyed. It's really strange. Okay, genotype ratio tells us how many offspring the Punnett square will be from each genotype. Genotype ratio follows the following format. Genotype ratio is always 1 to 2 to 1. 1 homozygous dominant, 1 homozygous recessive, 2 heterozygous. Phenotypic ratio tells us how it's going to appear, and it actually is going to show us this format. It's always 1 to 2 to 1. 1 to 2 to 1. This is red, that's I don't know what that one is, and blue. So this is actually a co-dominant kind of a thing where we get a red dominant, blue is recessive, and if we get one of each, it's co-dominant. It's a little bit of both, but basically it's the same ratio. It's a 1 to 2 to 1. Punnett squares. Pea plants, round seeds, dominant to wrinkled, cross 2 heterozygous. There's one of each. And in this one, we're going to get 2 R's, round seeds. And in this one, we're going to get an R, a dominant R and a recessive, round seeds. And this one, dominant R, recessive, round seeds. And in this one, we're going to get 2 recessive, wrinkled. So list the genotypic and phenotypic ratios. We have a 1 to 2 to 1 when we talk about genotype, 1, 2, 1. And phenotype, we actually have 3 round and 1 wrinkled. So we can't have them different. Which are purebreds? Purebred, purebred, hybrid, hybrid. Purebreds have the same, hybrids have different. Let's take a brown-eyed woman, has a father with blue eyes. That means that she's got an allele for that. And a woman marries a man with blue eyes. So if the couple were to have kids, what percentage of their children would have blue eyes? So there is the mom who had a blue-eyed dad, but she's got brown eyes. And there's the man, has blue eyes, is a purebred homozygous recessive. In this box, we're going to get brown. In this box, we'll get blue. In this box, we'll get brown. In this box, we'll get blue. So what's our ratio? Our percentage? It's a 50-50 chance. Which are purebreds? This one and this one. Which are hybrids? This one and this one. Monohybrid versus dihybrid cross. Monohybrids when we look at one trait, like color, size, shape. Hybrids, and this is an example of that. We're looking at one trait. Is the dominant carrier normal, or is the carrier dominant, which is normal, or is the carrier it has to be a carrier, so she's got to have the recessive, which is the damaged gene. But dihybrid cross are actually looking at 2 traits. There are trihybrid cross, which are 3 traits. But let me show you, if you know this one trait has 2 squares for each one, so 4 squares. This one actually has 4 squares for each one, which actually gives you a whole bunch. So if you take this heterozygous and this heterozygous and put them up, we have to put them in different places, and we end up with a whole bunch of others, which are purebred and which are hybrid. Do you see any purebred in there? Should be one right there. And I think that may be it. Because the rest of them all have a mixture. These are definitely hybrids, parents. So let's figure out how we do the gametes in a dihybrid cross. Let's say we have a round seed and we have a wrinkled seed. And we have a yellow seed and we have a yellow pea and a green pea, because we're talking about seeds for a pea plant. Heterozygous round, heterozygous yellow. So we actually have one of each for describing its shape and one of each describing its color. And how do we do it? We do something called FOIL. And this is keeping science fresh. Stupid. What this stands for is we take the first allele. So we take the R and this Y. First, outside, inside, last. So first allele, we take this R and that Y. So we get an R and a Y. Then we take the outside, which is this R and this Y. And then we take the inside, which are these two, this R and that Y. And then we take the last one, which is this R and this Y. And we get those alleles. So these are the ones we're going to actually use to set up our object. So we take the first, we take the outside, we take the inside, and we take the last, FOIL. And we do the same thing on this side. And then all we do is we cross them. We take this is Dad, so we take a big R, and it doesn't matter which one we take, they're both big, and we take the 2 big Y's and we put them in there. And then we're going to move down, because this is the way it seems to be moving. And then we have a big R, big Y, little y. And then we have a dominant, recessive, dominant, dominant. And then we have a dominant, recessive, dominant, recessive. Then we have a dominant, 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 recessive, dominant, dominant, recessive, recessive. Then we have a dominant, recessive, dominant, recessive. And then we have a dominant, recessive, recessive, recessive. And then we have a dominant, recessive, dominant, dominant. And then we have a dominant, and I'm looking here and then there. So we have a dominant, recessive, dominant, recessive. Then we have a recessive, recessive, dominant, dominant. Then we have a recessive, recessive, dominant, recessive. And then we have a dominant, recessive, dominant, recessive. And then we have a dominant, recessive, recessive, recessive. And then we have a recessive, recessive, dominant, recessive. And then we have a recessive, 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 recessive. So how do we set it up? We have 9 dominant dominants. Do you see them? It's one there, 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 there. 9. And then we have 3 dominant recessive. Do you see them? There and there and there. And then we have recessive dominant. We have 3 of those. And then we have 1 recessive recessive. And I think that's the stop. And I think that's the stop. And that's the end. Thank you for 8B. Take good notes. I think you guys know this one better than some of the other ones we've talked about this year.